We've already seen how the sequence of a polypeptide is important for its structure. And in this lecture, we're going to look at some techniques that can be used to confirm the identity of a purified protein before we begin to study its properties. We'll briefly examine the differences between the composition of amino acids in a protein versus the actual sequence or primary structure. And we'll see various techniques that can be used to either sequence the protein or identify it. And one of these techniques is mass spectrometry, and this can be used to identify a protein based on its mass. These are the supplemental readings in case you're interested in delving deeper into these topics. Now, when we talk about the composition of a protein, we're simply talking about all of the amino acids that are present. So if you were to hydrolyze every single peptide bond using strong acid, you could quantify the amount of each of the amino acids present, and this is typically what you would see for all of the proteins that are found in the SwissProt database. So on the right is a graph of the frequency of each of the amino acids, that's on the y-axis, and on the x-axis is each of the 20 amino acids. You can see that leucine is the most common over here on the left, while tryptophan and cysteine, these are very rare. Now, remember I asked you which amino acid you would eliminate in the thinker question when we were looking at amino acid properties? It probably wouldn't be something that occurs quite often or is found in small amounts because those might have very important properties within a protein. And so any of these amino acids that do have important properties, such as proline's ring, that's helping with those beta turns, maybe histidine side chain that can be protonated and deprotonated around physiological pH, as well as cysteine that forms those disulfide bonds, those might be amino acids that you wouldn't want to get rid of. Two amino acids you may want to look at are asparagine and glutamine, and compare their properties as an option. But really, we do need all 20 of these amino acids since many diseases show us that if you have a mutation, that can really affect the protein structure and its function. Frederick Sanger is someone you may have heard about based on his work in DNA sequencing, but he was also the first person to sequence a protein. And the protein that he sequenced was bovine insulin, which is shown below. Now, he could have simply sequenced the gene for insulin and then translated it using a codon table. However, many proteins are post-translationally modified, such as insulin, which has those two chains linked by disulfide bonds. So in order to sequence a protein, you really first need to remove these disulfide bonds and then separate the two strands, and then you could use Edmond degradation, which will sequence the amino acids from the N-terminus. We could then use other techniques such as carboxypeptidase to determine the amino acids starting at the C-terminus. So these results really convince skeptics that proteins do have unique amino acid sequences, and that it can be quite complicated when it comes to analyzing them and thinking about them. And really, this has helped move forward the field of looking at the genetic code and the complexity of how proteins are synthesized. So Edmond degradation is named after Per Edmond, and it involves a series of chemical reactions where the N-terminal amino acid is first labeled with a chemical called PITC, and then a low pH is used to break the peptide bond, creating this cyclic PTH derivative, and that's highlighted in yellow. This labeled amino acid residue, so PTH alanine in this example, is released and then separated and identified. That shortened peptide, so glycine, aspartate, phenylalanine, arginine, glycine, can then go through a second round of reactions in order to determine the next amino acid in the sequence, or that next glycine. 3 more rounds of Edmond degradation would then reveal the complete sequence of the original peptide. And this can be a very slow process, and it used to take you all morning to do 1 round, but we now have machines that can automate the process similar to what you see for DNA sequencing. There are limitations to how many amino acids you can sequence using Edmond degradation, and it's usually around 100 amino acids starting from the N-terminal end. But remember, many proteins can be quite a bit longer. We can also see that there are post-translational modifications that may block that N-terminus, so you may need to use other techniques. One way you can get around some of these limitations is to generate smaller fragments of your protein, but again, you would need to purify them and separate them out before you can do Edman degradation. And this is where proteases and other chemicals can come in handy. So remember, proteases are digestive enzymes that use water to cleave the polypeptide chains. And if we cleave them at specific peptide bonds along the chain, then we can generate these smaller fragments that we could sequence using Edman degradation. Here are some examples of commonly used reagents in the lab. Carboxypeptidase A and B both cleave from the end of the polypeptide chain, so the C-terminal end, with carboxypeptidase A being less specific and cleaves before any residue as long as it's not proline, arginine, or lysine. While carboxypeptidase B can cleave specifically before arginine or lysine, so those positively charged amino acids. Now, these enzymes do have active sites that are specific for these amino acids, forming non-covalent interactions so that they can recognize and bind and then cleave the peptide bonds on the N-terminal side of these residues. These last three cleave after the amino acids, so on the C-terminal side of the peptide bond, where trypsin will cleave after lysine and arginine, but only if proline isn't found in that peptide bond. So remember, the structure of proline has that ring, so it can create some issues when it comes to the recognition of these enzymes. And then chymotrypsin prefers larger, more bulky hydrophobic amino acids like tryptophan, tyrosine, phenylalanine, leucine, and methionine. Cyanogen bromide is a chemical that can cleave specifically on the carboxyl side of methionine when it's found in a peptide bond. So just make sure you know the differences between these reagents and where they cleave, because they are really important when it comes to fragmenting larger polypeptide chains into more manageable pieces. Let's do a little example where you have a 15 amino acid polypeptide, and you could just sequence it using Edman degradation, but let's say you treat it with two different proteases and then use Edman degradation. So if you first treat it with trypsin, you generate the following three peptides, and their sequences are shown below using their one-letter codes. But then you could also cleave the original polypeptide chain with chymotrypsin and generate the following four peptides. What is the actual sequence of the original polypeptide chain? And I'll give you a minute or two to sort of think about this, and you can also pause the lecture if you want to work on this on your own. In order to answer this question, you kind of have to think about it like a puzzle. So you have to remember that trypsin cleaves after lysine and arginines, or Ks and Rs, and chymotrypsin will cleave after phenylalanines, tryptophans, and the tyrosines that are present. When you cleave after these amino acids, that's going to leave them at the C-terminus of a fragment. One way you could start is to look for fragments that don't end in these amino acids. So this will be your C-terminus of the original peptide. So if we look, we actually see fragments that have histidines at the end of them. So this will be the end of the original polypeptide. You can then work your way backwards looking for overlapping fragments, or compare the longest fragments to those that aren't cleaved to maybe some of the shorter ones, and find how they overlap and match them up to form the original polypeptide. You can then check your answer by cleaving the original polypeptide with the different cleavage reagents. So trypsin would cleave after arginine and lysine to generate 3 fragments, whereas chymotrypsin is going to cleave after the 3 amino acids indicated in order to generate 4 fragments. So if we do have smaller fragments, we can sequence them using Edman degradation. But another way you can identify peptides is using SDS-PAGE, which we talked about, separating proteins on a gel, and then using a technique called immunoblotting or Western blotting that will identify peptides using specific sequences that are targeted by specific antibodies. Another way we can identify polypeptides is using mass spectrometry, and we'll take a look at how you can identify peptides based on their mass to charge ratio. This cartoon shows you the process of immunoblotting, where this first lane is kind of like one lane out of the SDS polyacrylamide gel that we saw in the last lecture, where we've got larger proteins up at the top and smaller proteins being separated and further down at the bottom of the gel. We would first transfer the proteins on that gel to a polymer sheet, and this is the blotting part of the experiment. Once you've done that, we first need to block any regions of the blot that don't contain proteins from the gel. So this is going to prevent non-specific binding of the antibodies, which, remember, themselves are proteins and could bind to the polymer sheet. The blot is then treated with a primary antibody, which would be specific for your protein of interest, and we would choose it or design it so that it only binds to a sequence found in your protein. You could then wash the blot and remove any unbound antibody. A secondary antibody is then added, and that's going to recognize your primary antibody. Secondary antibodies can be labeled in different ways, and the whole point is so that you can detect where these binding interactions are occurring. So if your protein is present, it'll show up on your Western blot, and we can also see how far it traveled down the gel. In this case, the secondary antibody is labeled with a fluorescent tag, and that's indicated by the yellow circle. And if you detect a band on the Western blot, then this will identify that your target protein is there based on its ability to interact with that specific antibody. Just to summarize, immunoblotting or Western blotting first involves the separation of proteins on an SDS-PAGE gel, and they are then transferred to a solid support membrane or a polymer sheet. After the membrane is treated with a blocking solution, we'll add some primary antibodies that recognize sequences of amino acids in your protein of interest. You'd wash any unbound antibody away, and then add a secondary antibody that recognizes the Fc domain of the primary antibody, and this secondary antibody will either contain a fluorescent marker or an enzyme that could generate a chemiluminescent product that you could see in order to detect your protein on that SDS-PAGE gel. Now, when a band migrates on the gel, it can also give you an idea of the size of the protein. So if it migrates further, it might be a smaller piece of your protein, for instance, if it's cleaved or modified, and you could also include beta-mercaptoethanol in your sample, and if you remember what this chemical does, it reduces disulfide bonds. So if we reduce those disulfide bonds and then separate your protein on an SDS-PAGE, those smaller subunits would migrate faster than the large overall protein that has quaternary structure held together by those disulfides. You can also use mass spectrometry to determine the molecular mass of a protein or peptides, as well as the sequence of a protein. So a mass spectrometer has 3 parts. It has an ion source, a mass analyzer, and then a detector. What happens in mass spec is that you'll hit your protein with a high intensity laser, and your protein will usually be found within a matrix that will contain metal and other small molecules to neutralize it. So that when you hit it with a high intensity laser or electron beam, we're going to ionize it, and then these molecules will become desorbed and travel through an electric field towards a negative plate through the mass analyzer. The detector will then detect them based on their time of flight, where highly charged molecules will travel faster, and it'll also detect based on size, so smaller ones will also fly faster. So their time of flight will really depend on their mass to charge ratio. And just on the top right is the type of data that you will see where we measure the intensity of detected peptides with the x-axis being their mass to charge ratio. Here's another example of what you can do to identify a protein in a cell. You could grind up your cells and obtain the soluble fraction of proteins, then run a gel to separate them and cut out or remove a band from that SDS page gel. You could then treat that band with trypsin, and remember trypsin cleaves after lysine and arginine residues, and then this will generate smaller fragments that you could separate based on maybe their hydrophobicity using reverse phase HPLC. You could then ionize them to generate ion peptides and analyze them by mass spectrometry. We could also do a second round of fragmentation using a neutral gas, and this would be called tandem mass spec because you have two in a row. And this will generate even smaller units like amino acids or peptides to get another spectrum of molecules. These spectrums are just showing you the relative abundance with smaller pieces on the left, where the x-axis is measuring your mass to charge ratio. And a longer sequence would generate a larger molecule, for instance, if we did have a peptide of alanine, glycine, and leucine. Now these letters on the peaks are just representing the different fragments where the start of the alphabet, so A, B, etc., would be for fragments that are made from the N terminus, while X, Y, and Z would be cleavage from the C terminus. So we can use tandem mass spec to determine the sequence of amino acids, where again, within the mass spectrometer, we're generating fragments of peptides based on the bombardment of inert gases and generating a family of product ions in which individual amino acids have been removed from one end. So in this example, the carboxy fragment of the cleaved polypeptide bonds are going to be ionized and analyzed. So the product ions will then be detected in the second mass analyzer, and the mass differences between the peaks will indicate the sequence of amino acids in the precursor ion. So remember, all of the amino acids will have different masses just based on the atoms that are present, and by automating this process and using computational tools, we can determine the sequence. So this is just a more complicated example of data that you might get, but it also shows how you can detect modifications like phosphorylation of these two serines. By adding phosphates onto these amino acids, you're going to change their mass, and mass spec can be used to detect these types of modifications. So don't worry, computers do all the work if you were to use this technique in a research lab, but the take-home message is that it's a great tool that can be used to identify and confirm the identity of your protein. So when it comes to sequencing proteins, people generally do use mass spec over Edmond degradation. So we've purified our protein and we've now confirmed that we have it in a tube, and really this is the protein that you want to study. Okay, so you hopefully have some sample left to do other things like study its structure, which we'll look at in the next lecture, as well as its function or ability to interact with other molecules. Now, the expression of a protein is something that we're not really going to cover in too much detail in the course, but a lot of research does study how a protein is made, particularly if there is a disease associated with it, in order to understand what happens along the way of making that functional protein. So the key take-home messages are that even though you've purified a protein, we really do need to identify what it is. And the first thing that you're going to do, that people usually do in a lab, is immunoblotting, where you can get antibodies that are specific for linear sequences in the protein in order to identify it. You can also send out your protein for mass spec, and that's going to identify it based on the unique mass of the atoms and the amino acids that are present. Or you can use Edmond degradation in order to sequence them from the N terminus, but remember that does have a limitation of about 100 amino acids. That's where we can use protease enzymes or other chemicals like cyanogen bromide in order to fragment these very long polypeptide chains into smaller pieces that could then be sequenced or identified through mass spec. So think about that question that we did, as well as a review of all of these techniques before doing the brain break questions and moving on to the next lecture.